Thanks. So most of what I'll talk about is really the main subtypes, the anaplastic large cell lymphoma, the AITLs, and the peripheral T-cell lymphomas, or the peripheral T-cell lymphoma NOSs. And it's a small group today, but this is a very important topic, and it's sort of near and dear to my heart. So it's really an honor just to present to you guys, and if there's, I think probably best to save the questions till the end, and we'll just go ahead and get started here. So the learning objectives of this talk, we want to identify encouraging treatment options for newly diagnosed peripheral T-cell lymphomas. And the peripheral T-cell lymphomas that mainly we talk about are those three subtypes: the sort of not otherwise specified group, the AITLs, and then the anaplastic large cell lymphomas. So we'll talk about some of these some of the newer studies that have been out, and talk about some of these encouraging treatment options. Then we'll talk about just briefly a little bit about clinical trials and sort of the importance of them. And then we'll talk about some of the ongoing studies, new clinical trials that are out there, and different encouraging therapies. So this is just kind of the overall outline, introduction, a little bit about clinical trials, talk about peripheral T-cell lymphomas and encouraging therapies there, and then we'll talk about the novel approaches and ongoing clinical trials, and then a little bit about my vision of where we'll be at in 2020. So as you guys know, overview of T and NK cell lymphomas. Often they're lumped together, and they're about 10 to 15% of non-Hodgkin lymphomas. So this is 80,000 per year. So that gives us about 8,000 new cases per year. So while it's rare, it is not. There's enough. There are plenty of them that pop up and need better treatment, certainly. So these diseases, while rare as a whole, they, as you see in this pie chart here, they get broken down into a number of different subtypes, and sometimes people will say there's more subtypes than patients, because they are a very heterogeneous disease, and there is some variability among the outcomes for these different subtypes. And the more and more we learn, the more and more it becomes more difficult to or more detailed in terms of our ability to subtype them and subclassify them, and ultimately better treatment options based on the subtype. And we'll talk a little bit about that. Again, these are the main subtypes we'll talk about. The angioimmunoblastic T-cell lymphomas they've sort of expanded in terms of what we used to think about as just the second line, the angioimmunoblastic T-cell lymphomas. We now have these other two subtypes which kind of look like the AITLs, and that's these follicular helper T-cell lymphomas, or the nodal peripheral T-cell lymphomas with a follicular helper phenotype. It's just a little bit of nomenclature, but things have changed a little bit, but those are the ones we'll focus on for this talk. Again, these in general have an aggressive course, regardless of which of the subtypes that you have, and they are very heterogeneous, though, in terms of what drives each of these diseases and ultimately how we're going to treat them. Really the way that we treat them in 2018, standard of care is with chemotherapy, usually some form of chemo, whether that's CHOP-like, or whether it's CHOP or another regimen called CHOEP or EPOC. Most people get 6 cycles of chemo, and then we think about adding a high-dose, ability to give people real high-dose chemotherapy in the form of a bone marrow transplant or autologous transplant. So this is kind of 2018 where we're at, and we are actually now, even in 2018, really starting to improve our ability to move beyond just that kind of standard chemo times 6 followed by transplant. We're really learning more about these diseases, and hopefully can figure out how best to treat them. So for example, these markers, these are what we call predictive biomarkers. These are all relevant to the anaplastic large cell lymphomas. So these are markers, the ALK, many people know ALK positive versus ALK negative anaplastic large cell lymphomas. But then we have these are what we call DUSP22 and TP63. These are markers within just the ALK negative anaplastic large cell lymphoma that are really helping us prognosticate and predict which are the more difficult to treat anaplastic large cell. So for example, patients with this DUSP22 mutation or rearrangement, so these are DUSP22 mutated ALK negative anaplastic large cell lymphomas. They do very well in comparison to people that have this other marker called TP63. And so we are learning that the management should probably not be the same for you if you're a DUSP22 positive versus a TP63 positive patient. So this is just, a really, really advancing kind of where we're at, even a handful of years ago. These are two other markers that we're learning are able to really subclassify within the peripheral T-cell lymphoma NOS group of patients. So these are what we call transcription factors that are important in peripheral T-cell lymphomas, are important into the development of these. And so we know that PTCL NOSs that have, say, this TBX21, this is the blue line, those patients do much better in general with chemotherapy in comparison to the GATA3 patients. And so that we're still, trying in the form of clinical trials and still trying to learn, if with standard of care we're doing so poorly with that pink line, how we can identify these patients at diagnosis and then ultimately potentially offer these patients clinical trials or if we know that the standard of care is so poor, we really need to be more diligent in trying to find better therapies for these patients. So these are kind of a number of markers. We have these other markers below the last four there. That CD30 we know is becoming very important, and we can talk about one of the new trials that's just being coming out and going to change the standard of care for patients that are CD30 positive. Epstein-Barr virus is a virus that's associated with some of these. And so we're thinking, we identify these markers and they help us predict sensitivity to certain therapies. And I'll kind of illustrate how that happens. So again, that we talk for a few minutes about this DUSP22 and TP63 story. So again, they predict DUSP22 positive patients do much better than the patients that are DUSP22 negative. And so there was a paper that was published last year showing that patients that are DUSP22 positive really probably don't need to undergo an auto transplant. So it spares people a lot of toxicity associated with bone marrow transplant in this subset of patients, and they still do very well even without getting a transplant, which is a good thing. Again, the story of EBV and Epstein-Barr virus, we showed that patients that have a sort of elevated viral load in their blood do very poorly, as you can see kind of this curve with the line. And so, we're in our group is particularly interested in trying to find therapies that may really benefit this patient population. And, we can easily identify this virus, and if there are ways we can use the virus to then target and treat the patients and have better outcomes, that's a lot better strategy than this one-size-fits-all approach. Again, so these are the therapies that we really have available and are FDA approved. So pralatrexate, brentuximab, romidepsin, and belinostat, those are all approved in relapsed peripheral T-cell lymphoma patients, with the exception of brentuximab only approved for CD30 positive ALCL. So one of the things that we've really learned that this class of drugs, the two drugs on your right side, the romidepsin and belinostat, Those are in a class of drugs called HDAC inhibitors. And we're learning that if you look at responses to these drugs, we see that the angioimmunoblastic T-cell lymphoma group really seems to respond more favorably than the other groups to this class of drugs, the HDAC inhibitors. Whereas, pralatrexate, maybe on the other hand, doesn't seem like it's quite as good of a drug for angioimmunoblastic T-cell lymphoma. And so, these are things that we're really learning. We've identified different mutations that these tumors have that you see up there, this IDH and TET2, and those are things that help us, again, be able to identify patients and hopefully identify a marker that we can then really treat people more strategically and better than we have in the past. Similarly, this drug azacitidine, while it's not approved, but it's been shown that patients with this particular DNMT3A mutation seem like they respond more favorably. The azacitidine is kind of a drug that's in a same category of what we call epigenetic drugs. So that's what we see here and that's what epigenetic modification is really what the romidepsin and belinostat do. And so these are just things that we're, now, 2018, really we know, and we do use these tools to help treat people now. Aside from identifying certain mutations and being able to predict, we actually know certain subtypes now don't respond at all to drugs like CHOP. This is a very rare subset of peripheral T-cell lymphomas. But in the recent NCCN guidelines that'll come out this year, we actually recommend that patients do not get CHOP, and actually one of the preferred option is giving them a different chemotherapy called ICE chemotherapy. And then our recommendation is really to undergo a different kind of transplant, the donor transplant afterwards. So these are just ways that we're really, improving options and improving our previous approach, which was, again, sort of a one-size-fits-all with CHOP. Now we're trying to really help tease out how we should treat people. So briefly, that was this is a little bit lighter in terms of talk a little about clinical trials in general, and then we'll talk about the trials that are available currently. I know this is probably a group that knows very much about clinical trials, so I just have a few slides, but just to go over really what a clinical trial is, because it is important and it's really the way that the field moves forward in clinical medicine. But a clinical trial is a prospective evaluation using human subjects. It's designed to answer a specific question about whether that's a therapeutic or an it's to ask a question about a certain intervention and whether it's the right option. So, for example, in clinical trials in peripheral T-cell lymphoma, we may ask the question, is romidepsin better than pralatrexate for angioimmunoblastic T-cell lymphoma patients? I think we probably know the answer to that based on the data that's there, but that's an example of a question. Unfortunately, drug development takes a long time, and it takes a long time to go through the various phases of clinical trials. And one of the even more challenging things in a rare disease is that these clinical trials take a long time to run because it just takes a long time to find all the patients. But this is actually on the x-axis there is years. So you see that from the time we discover a drug, it's a decade before we can really potentially even get it FDA approved, which is unfortunate and very long time. And the FDA does work hard to try to help speed these things along, but that's sort of where things are and how things can progress. The people that are involved in clinical trials, we have the sponsor, which is often a pharmaceutical company, but can be other players as well that can sponsor trials. The LRF can sponsor trials, for example. And investigator, that's me or someone else who's really running the trial at the site. And then there's a relationship that we work together to try to do what's best. These are the various sort of equipment that's involved. You have a clinical trial protocol that maybe you guys are familiar with. The IRB is an overseeing body that makes sure we're doing things safely and following good practice. The GCP, and then an IND is what you file to get a new drug up and running. So this is what an IND is, investigational new drug application. And so anytime we have a new agent that we're studying, you have to file an IND, or a new indication, you have to file a new IND. So these things all take a long time to get running, as you may guess. People talk, FDA sometimes gets a bad name, but they really are there to help out and make sure that the clinical trials that are run in the U.S. are safe and the right thing to do for patients. And so they're making sure, number one, first and foremost, that things are safe, but also they're assessing the quality of the science. And often, having gone through this process very recently, it is a back and forth often with the FDA to come up with what is a balance of safety and trying to help patients. Good clinical practice is something that we all follow. We all have a set of guidelines that clinical trials are held to, and this is why when we run clinical trials we can trust the results because we operate under a certain set of principles that are kind of this good clinical practice. So why would you consider a clinical trial? We talk a lot about clinical trials, but I think this is a way that we can offer potentially new therapies, potentially better therapies. It's the way that we advance care and advance the standard of care in all cancers. Of course, we always weigh the risks and benefits of these things. And again, it's important, if nothing else, if you don't remember anything else about clinical trials, it's just really important to understand what the difference between a phase 1 and phase 3 trial is, because that's it's really paramount that if you enroll in a trial, you want to know, hey, is this the first time this drug's been given to anyone ever, or is this something that's shown some data that's very encouraging and is likely to get sort of FDA approved. So phase 1 studies are really studies that we are testing the doses and testing safety. So phase 1 is a safety study. We're making sure that the drug that we're giving is not going to cause harm. Its actually main endpoint is not a efficacy goal. Of course, we want all of our phase 1 studies to be successful and show good results and be effective. But the way that it's designed is designed to from a safety standpoint. And then phase 2, on the other hand, is really looking at efficacy. And phase 3 is where you're comparing the standard to a new therapy. In peripheral T-cell lymphomas, although it's becoming more common now that we have phase 3 trials, it is very difficult to accrue, phase 3 large randomized studies in peripheral T-cell lymphoma. And all of those drugs that I showed before, the pralatrexate, belinostat, angioimmunoblastic T-cell lymphoma, pralatrexate, romidepsin, belinostat, all of those drugs were approved on phase 2 studies. So that can happen and does happen in rare diseases, but the gold standard is really a phase 3 study where we can compare the standard to something new and exciting. And so, of course, we want to talk about novel and all the exciting drugs that are out there, and that's kind of why you came here to this talk. And these novel treatment approaches, they kind of come in 4 flavors, at least how I break them up. So we have antibodies or antibody drugs. We have a class called what we call targeted drugs. Targeted drugs are different than antibodies, although they both are sort of going after a specific marker or pathway. We have immunotherapy, something that's very, very popular and in the news a lot, and then cellular therapy. And there's some overlap between immuno and cellular therapy. And so we'll talk a lot about these 4 areas in the next 15 minutes or so. So we have monoclonal antibodies. So monoclonal antibodies have, in the B cell world, have really changed the standard of care, and have really rituximab came out in the kind of early 90s and has really changed our approach in all B cell lymphomas. And essentially rituximab is combined with is the drug that is added to any newly diagnosed patient, except in the scenario of now we're having new drugs that are better than rituximab that we're starting to add in. But it's been a very exciting therapy for B cell lymphomas. Obviously, we're not talking about B cell lymphomas, we're talking about T cell lymphomas. And we have two monoclonal antibodies that are both in use in T cell diseases. So one is brentuximab, and that targets a marker called CD30, and alemtuzumab, which is a marker which targets a marker called CD52. So the way that these monoclonal antibodies work is they target a marker that's on the surface of the cancer cell. So I couldn't find a T cell, couldn't find a nice figure, but I found one for rituximab. Rituximab targets this marker called CD20, but for either brentuximab or alemtuzumab, you could substitute CD20 for CD30 or CD52, and this figure would be accurate. So these antibodies will latch on to the surface of the cancer cell because they see this marker called CD20, or in the case of the T cell, in case of brentuximab, they see CD30. In the case of alemtuzumab, they see CD52. And then it leads to a number of different mechanisms by which the cancer cells can then die. And there are really kind of 3 different mechanisms by which monoclonal antibodies work. One is really that it directly hits the cancer cell and causes it to die. The other one is via the immune system, and it activates a kind of a cascade called complement, or the complement cascade. It activates this part of our immune system, and then our immune system can then recognize the cancer cells as foreign and goes off and kills it. The other way is what we call is a different mechanism that is allowing this cancer cell to then be recognized by different cells in the body, and then it can target it. So these monoclonal antibodies, in the news we talk a lot about immunotherapy. The antibodies are still really a form of immunotherapy and the original immunotherapy, and that's because of these two mechanisms and the way that they work. It's not just linking onto the cancer cell and the cell dying. And in fact, these other 2 mechanisms where it's sort of telling the immune system that this is foreign and it's this bad cell to kill it are really probably the primary mechanisms by which these drugs work. Unfortunately, we only have 2 currently available monoclonal antibodies in T cell lymphomas, which are those 2 drugs listed there. In the B cell world, we have a slew of different markers, and there are a lot of reasons why we don't have more markers. One of the things about comparing B and T cell lymphoma, in B cell lymphomas, you can really live without your B cells and do pretty well. You can't live without T cells, and outside of these 2 markers, we haven't been able to identify really unique markers in T cell lymphoma that we can target with antibodies. And so that's a challenge that in T cell lymphoma we have not overcome yet. But needless to say, these are the CD30 and CD52 are good monoclonal antibodies that are out there, and we use sometimes. the drug brentuximab. So this is a kind of a picture of what the drug really looks like. So this Y shape up there is the antibody. Brentuximab actually is an antibody conjugated to a drug. And so these red balls here are the drug that it's attached to. And so what it does is, if you recall in the previous slide, that the antibody is sort of targeting that marker on the surface of the cancer cell, and then causing those ways that the cancer cell can die. What this drug does in brentuximab is it not only identifies that marker, but then delivers this payload of this drug, the red balls, into the cell and causes more direct cancer cell death. So it sort of maximizes the bottom mechanism, if you remember the figure I just showed, of causing immediate cell death to the cancer cells. And this is a picture of an anaplastic large cell lymphoma patient who was treated with brentuximab. So all of those dots, if you're not familiar with looking at PET scans, all the dots there are bad on the left-hand side. Those are all spots that this person had cancer in. And so this is before being treated with brentuximab and then after being treated with brentuximab on the right. And you see that really all of the cancer cells have gone away. It is normal to have the three areas that you see still lit up on the right side. That's just the bladder and kidneys. Those are normally what we call PET, are active on a PET scan. So that's normal. So this person on the right, as far as we can tell, is cancer-free after brentuximab, which is a great. So it's a great drug for this patient population that uniformly expresses this marker CD30. But again, not all peripheral T-cell lymphomas express CD30, and so we don't have good drugs like this for all peripheral T-cell lymphoma patients. However, we're really trying to develop new antibodies in peripheral T-cell lymphomas. The challenge lies in being able to find markers. All these markers that we have are on normal cells as well, not just the cancer cells. So while they're specific to a particular marker, they're not necessarily specific to just the cancer cells. So CD37, it's a marker that's on B and T cells. This is a clinical trial that's in development. This is just the drug, this AGS67E. It's rolls off the tongue, but it's an antibody similar to brentuximab. It links a drug to it, and that's this MMAE. It's the same actually drug that is used in brentuximab. So rather than targeting CD30 and delivering a drug via that antibody, this is CD37 has a conjugated to this MMAE drug, and we've seen some responses in an early phase 1 clinical trial in a number of different subsets of T-cell lymphoma. So a couple cutaneous T-cell lymphomas, as well as a couple of peripheral T-cell lymphomas, and this is something that's being further explored. Initially, this was really looking mostly in B-cell lymphomas, but now we've seen maybe a signal in the T-cell lymphoma patients, and this is something that's going to be explored further. Another strategy is we have antibodies that can target multiple things at the same time. So say this we call a bispecific antibody that's targeting both CD30 and CD16. And there's a reason that maybe you'd want to sort of combine those two. CD16 is it can be sort of brings near CD30 other cells that can then help make this drug be more effective. And so this is what we call a bispecific antibody. So it's just basically two different antibodies linked together and hopefully makes the therapy more effective. And that's a new drug called AFM13. So these are all antibodies and some of the newer antibodies that are being studied in T-cell lymphoma. When I talk about targeting lymphoma cells, we sometimes will say, monoclonal antibodies or antibody-directed therapy is, we're targeting their specific therapies. But there are other ways to really target the cancer cell that are not antibody targets. And this is a very elaborate, detailed big picture there that I won't obviously go through. But antibodies, the way that they work, rely on having a marker on the surface of the cell, of the cancer cell, that we can identify and go after. But as I mentioned before, in T-cell lymphomas, we don't have unique markers to target. And so that's a problem, because if we target markers that are like CD30, they're on the cancer cells, they're also on other cells as well in the body that could be important. And so another way that we think about doing this is we want to target intracellular pathways. And so this is, we want to identify and figure out what's making a cancer cell tick, and then, turn that light switch off. So I think this is, and there's a number of different strategies that we're looking at and really trying to develop, and this is just another form of targeted therapy. This is kind of a list of the various pathways that we are currently going after, and I'll show you some of the data that's out there on using some of these drugs. Some of the more popular, some of these PI3 kinase inhibitors, the second line up there, are really some encouraging data in that with that target. And so what these, all of these circles and squares up here, these are really ways that our cell sort of functions and tells the cell to do other things. And in cancer cells, sometimes they can take advantage of certain pathways that we have up there. And for example, in T-cell lymphoma, we think that this pathway, the PI3 kinase AKT pathway, is important. And so if we have a drug that can target just that specific pathway, then we can turn these cancer cells off. But these are pathways that are active in lots of cells in our body as well, not just cancer cells, but the cancer cells can use certain of these pathways, be more reliant on some of these pathways. And we think that, for the example, T-cell lymphomas, that many of them are really reliant on this PI3 kinase pathway to grow and to exist. Similar to mTOR, they have mTOR inhibitor drugs. This is along the same pathway as PI3 kinase, but different further downstream. There are other drugs that target proteins in the cell that can be important to and prevent cells from dying. So there's BET and BCL2. Those are drugs that target proteins that can then tell the cells when you target it to then say you should die. JAK-STAT and SYK and some of these other pathways are all things that are in T-cell lymphomas, very interesting pathways that are used and are very active in certain subsets of T-cell lymphoma. And the trick is ultimately going to be, not every peripheral T-cell lymphoma uses this mTOR pathway. And so it's identifying the patients, kind of more what I was saying before about identifying predictive markers, find what's making the cancer cell tick, and then target it and tell it to turn off. One of the exciting therapies that's out there in anaplastic large cell lymphomas, so these are the ALK positive anaplastic large cell lymphomas, is using drugs that are ALK inhibitors. So crizotinib has been around for a while and FDA approved in lung cancers. And we see that in ALK positive anaplastic large cell lymphomas that patients can really respond to this ALK inhibitor. And there's been a couple of studies, and these are small studies to this point, but encouraging, in that these are all going to be multiply relapsed patients that are getting on a clinical trial with, say, this drug crizotinib, and we've seen pretty good responses thus far. I mentioned before a little bit about PI3 kinase inhibitors. So we have developed a number of PI3 kinase inhibitors that are approved in B-cell lymphomas, certain B-cell lymphomas. There are certain types of PI3 kinase inhibitors that inhibit different, that are more effective in T-cell lymphomas, and those are the ones that we're really trying to go on and develop for T-cell lymphomas. So this is we call PI3 gamma delta. So many of the drugs that in B-cell lymphomas are really just affecting delta, and so gamma delta inhibition seems like it's a good strategy in T-cell lymphomas to really stop the T-cell in its track and hopefully die. So, like I said, multiple PI3 kinase inhibitors have shown therapeutic potential across a broad range of malignancies, but also in T-cell lymphoma especially. And I'll show you some of the data from our earlier trial. So this is a trial that we had and had enrolled 27 patients at this time. There's been a few more that have enrolled on since, but this is just 27 peripheral T-cell lymphoma patients. Unfortunately, the way that many of these targeted drugs work is that they sometimes take time to work, which is why a big goal is to maybe think about pairing them with other drugs to have a quicker effect. So in this study, we saw that a lot of patients had to come off the trial early, but ones that could stay on for a couple months actually did pretty well, and you could see really nice responses in a subset of these patients that went on. It's always, when you look at these figures, it's always better if you see more lines under 0. That's a good thing. So the ones that are above, those are patients that are progressing, patients that are responding and getting to complete response have 100, the purple line there. Those are patients that got into complete response, and many patients that had near complete responses as well. So this seems like it is active in peripheral T-cell lymphomas. Of course, the trick is identifying which peripheral T-cell lymphomas that we should use this in. Because again, we know that peripheral T-cell lymphomas are pretty heterogeneous, and as you know, one person's peripheral T-cell lymphoma is not the same as someone else's, and so we really need to be able to identify which drugs are going to work for which patients. So as I mentioned, there's been a number of patients that have had really durable responses to this drug. The top line, this patient's been on now for over a year and a half. That's actually my patient, and she's 90. So this shows you that this drug is pretty well tolerated, and she's doing very well. But you see here that there's a subset of these of peripheral T-cell lymphoma patients that are doing very well with this drug that's very well tolerated. There's some recent data that was published trying to be able to, again, identify who is going to respond to these drugs or this drug. And we've really had some encouraging data that we can identify these we call predictive biomarkers to know who is going to respond to PI3 kinase inhibitors. So that's a good thing. These are the number of PI3 kinase inhibitors that are currently in development for T-cell lymphomas. So duvelisib just recently approved for CLL and not approved yet in peripheral T-cell lymphoma or any T-cell lymphoma, but is a drug that is being studied further in peripheral T-cell lymphoma. Similarly, the slides that I showed you before, we're using this drug at the bottom there, the RP6530, which is now called tenalisib, is is very encouraging as well. All of these drugs are fairly similar in terms of how they work and efficacy. Another drug that is encouraging that targets a downstream effect that's a little bit further down within the cell, sort of beyond where the PI3 kinase inhibitors affect, is a drug called everolimus, which was published a couple of years ago. These are very small numbers of patients, 4 peripheral T-cell lymphomas, 2 anaplastic large cell lymphomas, and 1 AITL. But they're treated with a very tolerable dose of this drug that's, much easier to much less side effects in comparison to real strong chemotherapy. Similar to the story on PI3 kinase inhibitors, much easier to take and much less side effects. And again, we're trying to target these cancer cells sort of in what's making them tick. We know, this is just showing, this was a study that was open in a number of different types of lymphoma. And open in cutaneous T-cell lymphoma as well as peripheral T-cell lymphoma and sort of other T-cells which weren't specified. But you'll see that there are same kind of story, a subset of patients that are doing very well with these drugs. But the trick obviously is trying to identify, who these people are that are, on drug for 4 years and not progressing, and who the people are that are that it's not working in, and then being much more smart than we are now about then using these drugs. So this is a summary of some of the targeted clinical trials. So we have PI3 kinase inhibitors, these other 2 drugs, MCL1 and the other MDMX inhibitor. These are targeting proteins that help that when you target them, then tells the cancer cell to really die, which is a good thing. We want cancer cells to die. The other 2 drugs up there are a little bit different in terms of how they work. One is a retinoid. This one's open at MD Anderson. And there's another drug called ixazomib, which is a drug that's approved in multiple myeloma and targets a certain part of the cell that basically when you target it and kill it, the cancer cells will hopefully die as well. So these are all, good, exciting drugs that are available, or are not available, but ongoing and being studied in clinical trial. Immunotherapy, so this is, one of the things that our group is very interested in, very focused on, and particular interest of mine. So immunotherapy, we have an old drug called Revlimid, which is a drug that stimulates the immune system. It does many other things. There's a newer drug that's targets a certain marker called CD47, and I'll explain that. And then there are these big class of checkpoint blockade molecules and checkpoint blockade inhibitors that are classified by the marker that they're targeting: PD-1, PD-L1, LAG-3, CTLA-4. And essentially what these drugs are doing is that you have a cancer cell and you want your immune system to be able to recognize that cancer cell and then kill the cancer cell. So our body has its own way of doing these things, and the way that cancer cells can continue to grow is that they'll upregulate these markers that then make them invisible to the immune system. And so by giving a drug, we call them PD-1 inhibitors, you can then sort of tell the immune system gets revved up and active and then can go fight those cancer cells. And as it says here, it can we call release the brakes on the immune system to go after cancer cells. This is a little bit more detailed. It gets a little bit complicated in the T-cell lymphoma world because we're talking about T-cells killing other T-cells. But it does seem like it's an effective strategy. So this is what happens when the tumor cell is a T cell. But we know that these markers that can kind of make it invisible to the immune system are on the cancer cells. And when we can block them, we can make the immune system can then see the cancer cell and go after it. There are, again, these are just different checkpoint molecules that can be on the surface of the cancer cell. They can also be on normal cells floating around as well. And by blocking this pathway, it tells the normal cells to go after the cancer cell. This is a drug, this anti-CD47, is another way that we're sort of telling our immune system to go fight cancer. So the CD47 is we call the don't eat me signal for macrophages. Macrophages are a cell in the body that kind of wanders around and engulfs other bad things that are around, including cancer cells. And so when this CD47 is expressed, it basically doesn't allow the immune system and allow these cells, in particular the macrophages, to eat the cancer cell. So you see here CD47 being expressed on the cancer cell, and then using a drug that blocks this pathway then allows the cancer cell to sort of be recognized by macrophages and get eaten by them. And obviously when they're eaten, the cancer cell will die. Of course, everybody wants to talk about CAR T-cell therapy and cellular therapy in general. The original, cellular form of cellular therapy was really an allogeneic transplant or donor bone marrow transplant. And the problem with CAR T-cell therapy is very similar to problems that we have with developing antibodies in T-cell lymphoma. So really what a CAR T-cell is it uses the same technology that antibodies use, in that they're targeting a specific marker on the surface of this cancer cell, and then causing T cells to proliferate and T cells to really go after this marker. But unfortunately in T-cell lymphoma, we need T cells to live. We can't live without T cells. And unlike in the B-cell world, when we give people CAR T cells for B-cells, for a B-cell lymphoma, we essentially get rid of all of their B cells in their body, the cancer cells and the normal cells, but they can live without B cells. Can't live without T cells for very long. And so, there are a number of different strategies we're trying to develop. Obviously the easy thing is to find a unique target in T-cell lymphoma that we can go after. For example, CD30, there's a CD30 CAR T-cell, which is encouraging. Not 2 anaplastic large cell lymphoma patients on the trial that's been published so far. But there are other ways that we're thinking about how it might be possible to use the CAR T-cell therapy and CAR T-cell technology to really go after T-cell lymphoma, because it's been very, very, very exciting in the B-cell lymphoma world. So there is a CD4, we call CD4 as a marker on all of your CD4 positive T cells in the body. Most T-cell lymphomas are CD4 positive. And so there is a CAR that's in development that goes after this marker on the surface of the cancer cell called CD4. CD4 is on lots of your normal T cells, and you can't live without CD4 T cells. That's when you don't have CD4 T cells, that's really what happens in end-stage HIV, is patients don't have any T cells. So one of the thoughts is maybe we can use these CARs as a bridge, get rid of the cancer, eradicate it, but then use it as a bridge to maybe a donor transplant, where then we can use someone else's immune system, sort of replace your current immune system without T cells with a new immune system. We need to be able to turn off the CAR T cell or get rid of it to make this strategy successful. But that is a strategy that's out there. There's also things called, so rather than having a T cell that is your CAR, there are things called NK cells that we use as a CAR. There are a number of different strategies around cellular therapy. I wish that I could have more slides on CAR T-cell therapy or cellular therapy in T-cell lymphoma. We just are not there yet, but eventually we'll be. So combination therapy is really the last section that I'll talk about in terms of new and exciting treatments in T-cell lymphoma. Many of the problems that come up when you target specific pathways or target specific antibodies is that the cancer cell finds its way around that specific marker. So if you target, say, an anaplastic large cell lymphoma, target with a CD30 antibody, sometimes the cancer cell can then stop expressing CD30, and then obviously the drug's not going to work very well. Similarly, when you take drugs like, I showed the slide before, an ALK inhibitors with ALK-positive anaplastic large cell lymphoma, if you block that pathway, sometimes the cancer cell finds its way around that pathway and uses a different pathway. So we want to think about using strategies that maybe combine targeted therapy or combine different mechanisms to be able to get rid of the cancer cell. So an encouraging study that was, this is 2 years ago, presented at a big conference, is combining the drugs romidepsin and pralatrexate. So both of these drugs are approved by themselves as a single drug in relapsed refractory T-cell lymphoma. And so the thought is to combine the drugs to have more efficacy, and there is some preclinical data suggesting in mice that shows that this is the drugs may be a good synergistic combination. And so this is a study that was open across a number of different subsets of lymphoma, open in Colombia with Owen O'Connor's group, and they combined pralatrexate and romidepsin and saw really nice responses. The overall response rate was much higher than either drug by itself, and the complete response rate was much higher than either drug by itself. So this is an encouraging strategy, and I think one that is sticking around and being explored further. So this is another romidepsin again. This is romidepsin combination. So romidepsin sort of affects, it's called epigenetic modifier. The lenalidomide or Revlimid is an immunotherapy drug, and then the carfilzomib is kind of more of a targeted therapy. So they combine all of these, and saw really nice response rates of around 50% in a very pretty heavily pretreated patient population. But what they saw, and similar to some of the things we mentioned before about romidepsin and belinostat being more effective in certain subsets of peripheral T-cell lymphoma, they saw that all of the angioimmunoblastic patients responded to this combination. So maybe this isn't a strategy that's going to work in anaplastic large cell lymphoma or the peripheral T-cell lymphoma, NOSs, but it may be the best strategy in the AITL patients. This is a study that was using the PI3 kinase inhibitor in combination with either romidepsin or bortezomib. So targeted therapy with a different targeted therapy, sort of taking different strategies on trying to hit the cancer cell at two different areas, and saw some pretty nice responses in these patients presented last year. So this is the drug. So both of these drugs we think have been shown by themselves to be very effective in the angioimmunoblastics, and then combined together were really pretty great responses, with an overall response rate. Almost of the 8 patients, only 1 patient didn't respond, which is really encouraging when you see response rates like that. So other combinations, other options. So there's an ongoing study. Whenever you see the NCT number, that's just the number that you can look up on clinicaltrials.gov to look and find out where these things are at. A trial in combination with a chemotherapy drug called bendamustine, this CPI-613, which targets some of the proteins that we talked about before. Then this is immunotherapy, durvalumab, so it's a checkpoint blockade drug in combination with either of these, either pralatrexate or romidepsin or azacitidine. That's a study that's open on the East Coast as well. There's been a number of different. This is a study that's open. Jasmine Zane here, and this is a strategy that she's used, kind of a way to do an auto transplant that's a little bit different than the standard way that we go about doing it, where they link a molecule to it that then makes sort of emits some radiation, and it's been shown to be pretty effective as a strategy. So these are all of the currently upfront open clinical trials in bold for peripheral T-cell lymphomas. So we've talked about most of these drugs. The only one that does not is void of CHOP chemotherapy is the third one down. That's the Revlimid and romidepsin study that's just open for older patients. But this is, these are the trials that we've had before that are not in bold, and the one that has been really exciting that is being presented this year and has been shown to be better than CHOP in a randomized study is this regimen, this what we call brentuximab in combination with CHIP. So it's basically CHOP without the O. This is the data from the initial phase 1 study, which was very encouraging, with much better, long-term durable responses than you'd expect with CHOP alone. But again, this study was not. This was just the drugs by itself, and the conclusions, very high response rates. Essentially everyone responded, which is very impressive, and complete response rates near 90%. And they just recently released this report. They're going to present this at our big annual meeting at the end of the year. But it was a positive study showing that the brentuximab plus CHIP is better than CHOP, and really met all of its endpoints. So this is for CD30 positive peripheral T-cell lymphoma, and is going to change the standard of care. So it's very exciting. Another regimen that's I mentioned before, this full CHOP. This is pralatrexate in addition to CHOP. It's been published in its early on in its development, and but is up in Seattle. Andre Schustoff is really piloted this approach and working on exploring this further. But encouraging response rates, encouraging complete response rates. This is actually my trial. So this is combining a regimen that's kind of a CHOP-like regimen in combination with immunotherapy for newly diagnosed patients. So this is, we just opened the trial, very excited about it. But it's a standard of care chemotherapy in addition to that immunotherapy drug nivolumab. We think that there's a strong rationale to think why that chemotherapy in addition to an immunotherapy drug is a right strategy. So this is really my last 2 slides here. So I talk about what's the future look like in general, and this is the future, I think, of all lymphomas, but also T-cell lymphomas. We want to kind of harped on the idea that we want to be able to choose the right drug for patients. There's not a kind of a one-size-fits-all that's probably that's going to work, which makes it really difficult in rare diseases. We have lots of tools to be able to figure out how we identify the right drug, different kind of screening tests to identify different mutations that may predict response to certain drugs. But then there are ways that we can actually test the cancer cells and see what they're sensitive to. These are all things that are in development and not something that we use in clinical practice right now. But this is really going to be the future, I think. And then once we choose the right drug and we treat, we need better tools better at monitoring disease, and being able to with more sensitive tools, identify when it's gone away completely, and if it hasn't gone away, then find ways to really get rid of it. This is a slide that's from the NIH group, just kind of showing, we talk about amount of tumor DNA that we can detect in blood, and we see that at diagnosis, with the right tools, we can detect these cancer cells in patients' blood, and then we treat them, and the dotted line detection detection limit of sort of standard PET scans, CAT scans, it's below that threshold, but the cancer cells can still be around and obviously can come back, and that's why people can get into complete remission and it can still come back because our tools just aren't as good as we'd like. So being able to develop tools that can detect the cancer cells at lower levels, and when we get below a certain level, maybe we've probably maybe already reached our maximum ability to treat that cancer with whatever therapy we're doing, but maybe after we've got to the maximal response, then we need to add on a different type of therapy to really clear it out and get rid of it forever. And so this is my idea of how maybe we could do this, and which kind of hits on maybe a little bit why we think the strategy of immunotherapy and chemotherapy is the right strategy. But basically, you have a person that gets, say, standard chemotherapy, would kind of debulk their tumor and get it down to a certain minimal amount, but then maybe can use immune modulating drugs to then sort of mop up the job and be able to eventually keep it away forever. So that's really my vision of where we'll be in the future, and really why I think immune therapy is a great strategy and will be something that we're continuing to use in T-cell lymphomas. So these are all the lymphoma groups. Of course, the Lymphoma Research Foundation there, and other ones pertinent to more T-cell, that are more T-cell specific, the Cutaneous Lymphoma Foundation and the T-cell Leukemia Lymphoma Foundation. And then clinicaltrials.gov is where you can find any of these trials. You can search by the NCT number that's listed, or you can search by the drug or investigator. You can actually look based on location as well. So I think, so that's all I have.